So I have the honor to introduce Gordon today.
She offered to let me introduce myself.
Yeah, usually that doesn't go very well.
No, and then we never get to the talk.
Exactly. Okay, so Gordon got a BA in biological science from SUNY Binghamton. I didn't know that. It's kind of fun. So, and then a PhD from the University of Southern California. He then stayed in warm waters and went to the University of Hawaii as a postdoc, and he stayed there as a researcher afterwards until 1990, when Gordon joined the faculty here. But I guess by then it was still
MSRC.
MSRC.
Right.
Right? You know, with the changer or sticker, it's not. He is the head of the SoMAS Marine Science Division, so some of you have met him in that regard, but he's also the director of the NAno-Raman Molecular Imaging Laboratory, which we'll hear about today, and then we'll hear next week about some other cool applications that can be done with some of the instruments here. Gordon is a microbial ecologist and microbial oceanographer. Lots of things, water column. He still goes on a lot of research expeditions. Not anymore.
Not so much.
But yeah, we're really excited that he's stepping in and giving this talk today. If you have any questions regarding the instruments afterwards, right, come talk to him. Bring your samples.
Yeah, please.
Yeah, thank you, Gordon. Yeah, you're still just on the top.
Okay. Live?
Yep.
Fantastic. Well, it's my unexpected pleasure to be talking to you today and telling you a little bit about what we've been doing over, turns out now, the last decade. We created this facility here in 2014. Okay, so can we get that talking thing out of the way? Maybe down there. Is that okay?
Yes.
Okay, so first I want to talk about motivation. A lot of the work that's been going on in my lab over the last decade. And it has to do with better understanding plankton community dynamics. This lovely figure that Alex Warden published a few years back shows basically the planktonic food web with all its complexity, and I think we all know by now that the planktonic food web is what drives the biological carbon pump in the ocean. But there's a lot of subtlety within the planktonic food web that we still don't understand, and we're largely, our understanding is limited by the techniques that we use. So there are all kinds of symbioses that we don't understand. There are localized activity around phytoplankton, referred to as the phycosphere. There are organisms, and we're identifying more and more, that switch their types of metabolism from being a photosynthetic organism to a heterotrophic or predatory organism. And these are all mostly unicellular organisms. There are saprophytic fungal-like organisms that Jackie Collier studies that not many people in the world know much about. There's parasitism and there's predation. All these processes are often not resolved because of the types of techniques we use. We use mostly community level measurements. So the motivation for studying the microcosmos are for the following reasons. So the traditional biogeochemical measurements that we typically employ and have for the last 50 years are really looking at net transformations that are at scales that are irrelevant to unicellular organisms. Most biogeochemical processes are dependent on microstructure and cell-to-cell interactions. And so many of the techniques we use are blind to these processes. So we come to recognize the importance of microspatial seascape ecology. What's happening down at dimensions smaller than a millimeter, down to micrometer. So this is what we're calling the microcosmos. And I'll come back to this in a moment, but for most microorganisms, a bacterium or a phytoplankton, their whole lifetime, their whole universe is encapsulated in 1 cubic centimeter. That's all they ever see or experience. Okay. And part and parcel with understanding the seascape is the idea that we need to better understand biogeochemical functions and relate that to phylogenetic identities, so that with all the genomic information that's coming out these days, we need to make better grounded inferences about what that genomic information means. So our current techniques are very poor at looking at variability within populations, meaning all members of a single species, as well as interactions among different species. Our techniques tend to overlook cryptic exchange of materials, whether it's carbon or sulfur or even oxygen. Our current techniques aren't very good at allowing us to see the deeper mechanistic underpinnings of these processes. So not too long ago, our concept of the way the ocean looked for microorganisms, where they were just sort of homogeneously distributed in the blue ocean, and on closer examination, we know that's not the case. And here's this artistic rendering that was published a while ago from Brook that is maybe a more accurate depiction of what's going on. And in our own lab, we look at the microspatial heterogeneity of bacteria. Each one of these little fluorescent blue things is a bacterium. And there's all kinds of organic particles around which they aggregate. And those can be sites of really intensive activity. So I really like this 1 cubic centimeter of seawater that Bill and Stocker published a while back. And it shows how much is going on in that very, very small volume of water, and how heterogeneous the processes and the distribution of organisms are. Biological carbon pump is driven by the vertical transport of biogenic material, organic material, which are again shown in cartoon fashion, another Raman Stocker illustration. And basically, it's illustrating that organic matter and nutrients can be highly focused around particles or around cells, phytoplankton cells that may be lysing because they're attacked by viruses, or autolysing for other reasons. And not only is there microspatial heterogeneity in the distribution of nutrients elements, but also many particles are actually have internally anoxic regions. So type of niche that's available is different than the surrounding waters. It can enhance growth of anaerobic organisms. Okay, so one of the big challenges that we've always had in marine microbiology is that most organisms don't care to live in our lab. We have difficulty cultivating marine bacteria. Phytoplankton, many we can cultivate pretty reliably, but it's estimated that 95% of all the bacterial species out in the ocean won't live in the lab, cannot get them to grow. And so the way forward is to develop culture independent techniques. And so there's been a big drive over the last 30 or so years to develop a variety of culture independent techniques. Many of them involve using stable isotopic probing or radio tracers. And there are a number of techniques that can look at activity of cells, individual cells rather than in total volume. And the two techniques that I'm going to be talking about and showing you today are atomic force microscopy to a limited extent, and stable isotopic probing, SIP, Raman fish, which is has nothing to do with scales or fins. It's fluorescent in situ hybridization. So the use of phylogenetic probes to identify organisms under the microscope. Okay, so first of all, what is Raman scattering? Well, it's not this. My favorite gag. It's not this, and it's spelled Raman after the fellow that discovered it, not Ramen. Okay. So what Raman scattering is basically a type of vibrational spectroscopy, complementary to infrared spectroscopy, if you've heard of that. And essentially, a sample is illuminated with laser light, which by definition is monochromatic and has one wavelength. All photons have the same energy. And when photons collide with the target, meaning a molecule, most of the photons are scattered at the same wavelength. That's the Rayleigh scattering. Notice the same color, same wavelength, same frequency. That's nu sub-zero. That's the frequency of the laser. But 1 in a million, 1 in 10 million of those photons actually gives up a tiny bit of its energy to cause the vibration, stretching and collapse of molecular bonds, and scatters a photon with lower energy. And the energy difference between the Rayleigh and the scattered is called the Stokes scattered emissions. And its energy, it provides information on what kind of molecular bond that photon struck. So we can generate a spectrum that looks like this, where in this case, the Rayleigh line is in the center here. So that's the laser light. And these peaks are the scattered emissions at different energies, which are attributable to different molecular bonds, different elements bound together. And there are all kinds of vibrational modes that we can look at. I got to get this out of the way. Go. And they provide us with information. Now, this technique, before I go into this technique can be used for all kinds of materials. We're using it a lot for microbiology and even looking at tissues of higher organisms. But it can be used in material science. This technique can be used in pure chemistry. It can be used in geological determinations. Okay, so it is a weak phenomenon. Again, a very, very small number of photons get Raman scattered. Most of them are Rayleigh scattered. So you need instruments that are really, really very, very sensitive and have really precise optics to separate the photons. Okay, so the instrument measures the change in light frequency after energy has been transferred to the target. And basically from spectra, we can determine very subtle chemical differences between either spots in a sample or different types of samples. And when we look at all the peaks collectively, it gives us some idea about the identity of target. So I'm just showing an example here. We look at one spectrum that came from an individual bacterium, and you see all these peaks along here. And each one of those peaks, or most of those peaks, we can identify as coming from nucleic acids, from proteins, from lipids, carbohydrates, and so on. So it's very, very useful in biology, but underutilized. There aren't a lot of biological labs that actually use this approach. Okay, so as I said, the facility is now 10 years old. We established it with a major research instrumentation grant from National Science Foundation in 2014. If you want to see it, it's just down the hall in Endeavor 170. And here it is. This is the Raman Microspectrophotometer. It's on an optical bench, which is floating on airbags, because it's very susceptible to vibration. And parked right next to it is an atomic force microscope. This is the working head of the atomic force microscope, which I'll get to in a moment. And the way we configured this is it allowed us to not only do Raman spectroscopy or atomic force microscopy independently, we have them, they can be optically coupled, which I'll show you in a second. So the entire schematic of the entire system is shown here, where we have 4 different lasers, actually 3 lasers, one that produces 2 different lines, so we can use different excitation energy, and then it's focused down to a basically an adopted or converted microscope that can be focused down to spots that are on the order of just a few tenths of a micron. Okay. So more often than not, we're just doing the Raman, but there are some applications where we redirect the laser light onto the atomic force microscopy stage, and we can then improve our spatial resolution by a factor of 10. We can actually get information from spots as small as about 20 nanometers. 20 nanometers. Okay. So what's atomic force microscopy? Well, it's a type of scanning. It's a scanning probe microscopy. It has horizontal resolution of 0.4 of a nanometer. It has vertical resolution of 1/10 angstrom. A human hair is 1,000,000 angstroms in diameter. The average human hair is 1,000,000 angstroms, just for reference. Okay, so when they say atomic force microscopy, we're really talking about visualizing things down close to the atomic level. Okay, so this is a schematic of how it works, rather than it being based on optics or based on electrons, like electron microscopy. It's based on contact. There's a tip that's 10 nanometers diameter at the tip, and the sample sits on a stage, and the stage rasters back and forth, and the tip moves up and down. It's just barely making contact with the outer electron shell of what's ever at the surface. And as the cantilever moves up and down, it reflects a laser light to a photodetector. And then the change in height or position of that light beam in the photodetector gets recorded. And we produce images that look like this. So that, if I didn't tell you that that was produced by an atomic force microscope, you might have said a scanning electron micrograph. But the instrument that we have down the hall can produce images like this. And this is actually a mouse cell produced by AFM. And we can operate either in a static mode where the tip is just riding across, or in a vibrational mode where it's oscillating. And that tells us something about the mechanical strength of the surface. So we get topographic information, and we can get mechanical information from the surface. These are some images that an atomic force microscope can produce compared to what a scanning electron micrograph produces. So AFM, this is the shell of a diatom frustule. You all know about diatom frustules made of glass, silicon dioxide, right? So AFM produced these images in parallel, the same organism. SEM produced those images. And right down the hall, we produce this image of the shell of a diatom here, where it's highly magnified. We're just looking at a 10 micron span of that diatom frustule. Okay, scanning electron microscopy is labor intensive. You have to remove all salts from the sample. You have to dehydrate the sample. You have to sputter coat the sample with a metal, a vaporized metal, and then you can get an SEM. The AFMs, we can do it under normal lab conditions. conditions that we're sitting in right now. SEM is also done under high vacuum, big expensive instrument. AFM we can do in this room if we were to choose. And we even have an accessory where we can do it underwater. So we can look at fully hydrated materials and get atomic force microscopic images. And here are some images of that Tanya produced. She's back in the audience there. These are that's double-stranded DNA. We're imaging strands of DNA that have been plated out on a mica surface. And I'll remind you that DNA is about 20 angstroms in diameter. These are artificial plasmids, again, DNA that we produce in a lab, not in our lab, but were analyzed in our lab. So we can image things that are really, really small, right? DNA molecules. Okay, so what I'm going to do now is go through basically 3 vignettes, 3 research projects that demonstrate the at least the biological capabilities of this instrumentation. Plug next week, Luis Medina will be talking about applying Raman to looking at microplastics out on the ocean. You'll get more of a exposure to some of the other applications there. One of the first things we did is ask the question, can we measure single cell growth rates of photoautotropic organisms? And we started with a study using Synechococcus, which is one of the most common cyanobacteria in the ocean. And basically, these are stable isotopic probing experiments. And the way we set it up is we had a control, which is just natural abundance of C13, which is about 1% of all the inorganic carbon. And then we enriched 11%, 20, and so on. And then we incubated replicate cultures with these different enrichments. And what I'm just showing here are growth curves that are based on just in vivo fluorescence of the organisms. And you can see with all treatments, there's no difference between the enriched cultures and the control. They're all growing at the same rate. They're statistically indistinguishable. And this is just a fluorescent, autofluorescent micrograph of those little Synechococcus cells that are just a couple of microns, 1.5, 2 microns in diameter. So if we follow their growth through time and we look at the carbon-13 labeling, basically at the beginning of the experiment, they're 1% C13. After 1 generation, and if they're growing in 100% C13 bicarbonate, 49% enrichment. After 2 generations, 74, and so on. So basically, if we look at generations, so each generation means a doubling of the population, okay, and then the percent of C13 in the biomass, we can predict how much labeling there will be, and when we hit that inflection point, when they're basically approaching saturation. We looked at, again, control and highly enriched media, and we looked at individual cells. So on the instrument, we can either get a right field microscopic image of the cells or a fluorescent image, which is shown here. And essentially, what we're looking at, the top spectrum is the control, where there's been no enrichment whatsoever. And we see 3 distinctive peaks that are emanating from carotenoid pigments that are in the cyanobacteria. And then if we look at days 5 through 9 in the 96% enrichment, what we see are frequency shifts. You know, at first it's very subtle, and then we can see it after day 6, day 8, day 9, that all the frequencies are shifting to your left. And we call that red shifting because it's lower frequency, it's a warmer color, red shifting. Okay, so what was interesting about this comparison is that during these time course, which we extended further, we couldn't see community growth. We couldn't see population growth by doing in vivo fluorescence, which is a fairly sensitive technique. But in the first few days, looking at single cells with Raman, we could actually detect incorporation. So we knew the cells were growing even though the fluorescence wasn't revealing that there was any growth. Okay, so how do we interpret these spectra? Imagine a diatomic model here where we've got two carbon atoms bound to one another, a single bond. If they're both C12, they'll have one characteristic frequency. If one of the atoms is C12 and the other C13, they'll have an intermediate frequency. And if both atoms are C13, they're going to have a slower frequency. They're heavier, and so they will vibrate slower. So this is rather remarkable that with an optical technique, we can see the effect of one neutron being added to each atom. Okay, so in this case, this is the spectrum for, it's just a portion of the spectrum. It's looking at the carotenoid, and almost all of the curve is under this higher frequency at the, I can't remember what it is, 1535 or something like that, with very little contribution of the C12, C13. As we, when we add labels, the media is 54% enriched in C13, and after 3 days, we now see that peak changes shape, and when we deconvolute the curve and we look at the individual peaks that are making up that complex curve, we can then see the contribution of the C13, C12 here. And as we get even further along, go down to the bottom here after 9 days, there's a distinct peak for C13, C13, the C12, C13, the C12, C12, and then derive what percent labeling has occurred. We can get what the isotopic enrichment is. Okay, so in this study, again, we were just looking at carotenoids, and basically we looked at 3 different wavelengths, and what you see over time is the frequencies are going down, that red shifting that I mentioned before, red shift, red shift. In the control, there's no change, and we have the dotted line that you can barely see because it coincides with the regression. Prediction is how heavy should they get based on the growth rates calculated from the fluorescence measurements, and they overlap completely. When we look at a real an extensive range of isotopic enrichments and time points, we see that there's a very good prediction for the change is proportional to the fractional abundance of C13 F sub cell. That's the fractional abundance or percent of C13 that's in the cells. So we can predict that very accurately. Don't worry about the math, but basically what I'm going to tell you is from those from that this relationship, we can calculate the growth rate of every single cell that we interrogate. And in this study, it took us 2 seconds per cell to look at each cell. So we looked at hundreds and hundreds and hundreds of them. And we can calculate the growth rate of each individual. We also asked the question if there are organisms that have different levels of enrichment and different growth rates, and we mix them together, can we recognize them under the microscope? And that's what this image is showing us. Each one of those fluorescent objects is a cyanobacterium in this culture. And we, in parentheses, is the population growth rate determined by fluorescence, or you can do by looking at the accumulation of cells. And the value to the left of that is the single cell measurement derived from Raman. Okay, and we see that there is a really good correspondence between what we would predict from population and what we observe from cell to cell. Okay, so takeaways from this, one of the things that surprised us initially is that we were working with a clonal culture of Pseudococcus, meaning that all the cells came from a single parent at some point in history, and they're all living under the same conditions in a flask with supposedly uniform conditions. But there was cell-to-cell variability in their growth rates, and so coefficient of variation in the Pseudococcus was about 27%. So there was fairly significant variability. When we did the same experiment with a diatom, Thalassocyra, its coefficient of variation was a little bit lower. And then we searched the literature, and there's probably now close to a dozen papers maybe that have been published that have used different techniques to do single cell growth rates. And they've reported variability on the order of 19 to 51%. So our measurements we're convinced are real, they're not analytical error. And it's just that within a given population, even subjected to the same environmental conditions, you can have a range of growth phenotypes that are in that, even though they, at some point in history, they came from one individual parent cell. So under these conditions with a uniform population, what does that tell us about what's happening out there in nature, where you've got heterogeneous population with multiple species within a community? It's a lot of variability. And this little cartoon basically illustrates, if you do a community measurement, you get one value and you don't know anything about outliers. But if you do single cell analysis, you find that you have a number of phenotypes, and that can actually provide useful information for predictions of how the community will respond. Okay, quickly move on to looking at phytoplankton mortality by viral infection. This is a study that published in 2021. It's primarily the work of Elena, who I believe is here. And our study organism, the host, was Amelia huxleyi, which is a coccolithophorid, which forms blooms every spring that you can see from outer space because of their white calcified tests. And they are super important in carbon export in many regions of the ocean. And this species is cosmopolitan. It's found in all oceans from the tropics to the poles. So in collaboration with a colleague up at Bigelow Lab, we got hold of a virus that attacks P. hux. And here it is. These things that look like popcorn are actually, this is an AFM image of the viruses isolated from Amelia huxleyi. Here's a blow up of one individual, and here's a transmission electron micrograph of an individual virus. And these viruses are about 160 nanometers in diameter. That's pretty tiny, right? Very small. And they're an unusual virus for phytoplankton because they have an envelope around them, a lipid envelope around a protein capsid. Okay, we asked the question, can we actually get Raman information from these 160 nanometer particles? And to our delight, the answer is yes, we can. So here's the target. Here's the Raman spectrum. And I'll just point out, this is from a single virion. And these peaks are indicative of protein, DNA, and lipids. As I mentioned, they're enveloped, so they have a lipid envelope. So this is from a single virion. This is average of 5 virions. This is an average of 20. The shaded area around that, the black line is the average. The shaded area is the standard deviation at each pixel. And you can see it, the measurements are really quite reproducible. Okay, well, we surveyed a number of viruses, and by Raman spectroscopy. And so a complete virion again looks kind of like this. The background is essentially flat line, no information there. That's good. We like that. But on occasion, we would find what looked like ghost viruses. This image here is basically, it's either an empty envelope or a cell vesicle, we're not sure which, but we can see that spectroscopically it's different from complete virion. And what's missing are the peaks at 724 and 782, which are DNA. So we can see that there's no DNA present there. So that was kind of a neat discovery. Then we asked if we have isotopically enriched hosts, does the virus inherit that same isotopic signature? Well, one would expect yes. Yes is the answer, but can you actually demonstrate it? And we did. So this is the Huxley host. The bottom is the control. The top is the label. We're going to focus in on this area of the spectrum. Unlabeled, there's just one big peak here, which is due to the amino acid phenylalanine. In EHUX that was incubated with enriched media that was like 60%, 65% enriched, we see multiple peaks. The big one has shrunk basically, and we see 3 other peaks, and those are attributable to different levels of labeling of the phenylalanine, which is a 6-carbon ring. Then we isolated the viruses and we took the average value on the viruses. So here's the control virus, there's its phenylalanine peak. Here's the experimental viruses, and there are again 4 peaks. When we calculate the percent enrichment, the virus shows the same degree of enrichment as the host cells. So it's getting up. I mean, when we predict this, they're getting all their carbon from their host. Makes total sense, but there's potential for a variety of experiments with this kind of approach. Another thing that we learned quite by accident is that we could do we knew we could do 2-dimensional mapping, where the instrument scans and stops every 1 micron and takes a spectrum. So we can do 2D scanning and then just map the distribution of a given peak that's diagnostic for something. So in this case, we have what we're calling protein portraits and DNA portraits of the same cell. So these come from the same cell. Here's an uninfected cell, a lot of nuclear DNA concentrated. Here's the cell itself. And then after a day after infection, lots of protein, the DNA signature is getting faint and more diffuse. 2 days after infection, a lot of the DNA is already gone, and it's likely that the host is shedding the viruses that are killing it and taking with it DNA. So we can visualize this in 2 dimensions. Okay. All right. The last vignette was primarily the work of Felix Weber. He's back in Germany, and he was supported by a Moore Foundation grant working with me. And basically, he wanted to know whether everything I showed you with the Synechococcus, that's with photoautotrophs, so they're photosynthetic. Can we do similar things with heterotrophic bacteria? And not only look at growth of bacteria, but death as well by predation. So we did a series of experiments that basically went like this. We had unenriched media where we grew the prey, enriched media in which we grew the prey. We measured their growth rates by spectrophotometry, growth rates of the entire population, and we looked at single cell growth rates by Raman measurements. This clone, the strain of E. coli that we use, is kind of neat because we can stop it from growing by brief exposure to ultraviolet light. So we can grow it to a point where we say, all right, stop growing. Now we're going to feed you to the wolves. Now we're going to feed you to some protozoa. So we can stop their growth. And Basically, we had azenic cultures of a ciliated protozoan, tetrahymena, very well-studied system. And we can grow this in the absence of bacteria, just giving it sterile nutrients. And then basically, in the control, isotopically light prey, they were allowed, they were eaten by the tetrahymena, and then isotopically enriched, they were also eaten by tetrahymena. We did microscopically cell counts of both partners in this relationship, and then we did single cell analysis by Raman. Just real briefly, again, the isotopic enrichment of the media did not change the growth rates of E. coli, so this is in isotopically light blue, isotopically enriched. This is just spectroscopically determined growth rates of the entire population. And then in the Raman, what we did is we focused on the region of the spectrum that gives us the vibrational modes from phenylalanine. So P spectrum means phenylalanine, again, essential amino acid. And this just shows that when cells are isotopically enriched, we get multiple peaks, and the heavier they get, the bigger the peak to the left becomes. Okay. So we did a time course, and we could watch at the beginning, it's all C12, and then through time, we see that we lose that C12 signature almost completely, and we get a C13 signature. So again, we can calculate how enriched they are and how fast they're growing. And yeah, I'm not gonna need to go through this. Basically, the same arguments that we made for the photoautotrophs. By measuring each cell's isotopic enrichment, we can calculate each cell's growth rate. And when we compare each cell's growth rate with growth rates that were determined from for the population by regular visible light spectroscopy, we see that there's no statistical difference in Raman determined measurements versus which are single cell versus population. Okay, so then the next step was the feeding experiment. So we're feeding tetrahymena, these heavy bacteria, and here this is an image of tetrahymena, and I'm pointing to the food vacuoles. If you look closely, you can see a bunch of little bacteria in there, all clumped up. They've been phagocytosed from the environment, and they're being digested. So you can see vacuoles at different levels of digestion in this. All right, so it's the same story as the previous slide. Through time, we find tetrahymena individual cells red shifting their phenylalanine peak, getting heavier and heavier. And so from this, we can calculate how much they're eating and how fast they're growing. That's pretty cool. But the other thing we can do is, again, two-dimensional mapping. And early in the grazing experiment, so warm colors mean heavily enriched with C13, cool colors mean isotopically light, okay? And we see hot spots where the color's warming up, and this is at 6 hours, only 6 hours into the experiment. And so these areas with warmer color, those are the food vacuoles, because we're seeing the signature of the prey. And then as time goes on, and we're a little over a day, that carbon-13 is basically uniformly distributed throughout the biomass of that one cell. And we can see the cell after cell after cell. Okay. So these 2D maps, basically, it's 400 spectra per map in this case. Each spectrum took 15 seconds. So it took about 3 hours to produce an image like this. So we asked, 2D is really cool, it's neat, but you're not really seeing the total in vivo distribution of the organelles or the isotope. Can we look at cells that have never been dehydrated? Can we look at them totally hydrated? And the answer, yeah, we can. And basically we have one of our lenses is 63x water immersion. And so Felix was able to get these ciliates, never been dehydrated, just preserved with formalin, to stick to a stainless steel slide underwater, and overnight got a three-dimensional map. And this is basically a description of how we did it. We looked at, we set up a grid, and I forget what the resolution was, every few micrometers, would stop and take a spectrum, stop and take a spectrum. And we did this in 7 planes in the vertical. And then the software just puts that all together for us and forms a three-dimensional image. So these were not isotopically labeled. This was just proof of concept. Can we do it? And so we get spectra where we can see signature for DNA, protein, lipids, and then we look at it in 3 dimensions. And the software is pretty darn cool because we can look at the cells from any angle. We can slice through it in all 3 dimensions. So the blue is the distribution of protein in this protozoan cell, and the red is the distribution of DNA. And if you were paying attention, you might have noticed that the DNA is present in multiple sites here. And that's because ciliates have multiple nuclei. They have one big macronucleus, and then they have multiple micronuclei. So we thought that was pretty neat. Oh, all right. So to sum up, the sample prep requirements for doing Raman analysis are really relaxed compared to many other types of techniques. We can look at live stuff. We can look at dried stuff, preserved stuff. We can look at stuff underwater. Frozen cells we can look at. So there aren't these really stringent sample preparation requirements to get really useful information. And by analyzing Raman spectra, we can quantitatively determine isotopic enrichment. And we use C13 here, but my student Natasha is using deuterium, and we've used other isotopes as well to trace different processes. It's an approach where you can look at cell by cell, how much variation is there in the response of an organism to a particular perturbation or stimulation. And we can combine the SIP-Raman approach with phylogenetic probes. So when we're out in nature, we can ask the question, who am I looking at? All bacteria under a microscope look kind of alike. But with a phylogenetic probe, we can say that's a member of the Delta proteobacteria clade, and it's doing this process, and this is how fast it's doing that process, cell by cell. Okay, so I would advocate that 2 tools that we have down the hall provide a unique way to look, view the microcosmos and look at a lot of these different processes that are giving ecosystem modelers all kinds of headaches, like carbon flow between symbionts, carbon flow between predators, et cetera. We can look at them on an individual basis. And with that, acknowledgements, the instrument came from the National Science Foundation. A lot of the science was supported by the Moore Foundation, even more by other NSF grants. And just to acknowledge Team Narmal, Tanya, Elena, Elizabeth, or Liz, Luis, Felix. Steve's not a member. He provides all the mathematical background for us. Student that graduated, Ashley, and current student, Natasha. Okay, so thank you for your attention.
Our students for today are Zach. Where's Zach?
Yeah.
Well, you're in the hot seat, huh?
Yeah, so when you were talking about the actual Raman device, you said it was really sensitive to vibrations, and so it had that like lower plate. In your time in this lab, have you ever experienced like really bad like construction nearby that kind of made the vibration like I like it went through the plate or like made it
Yeah.
worse.
We haven't, have we, Tanya? Yeah, do you recall? Yeah, so the optical bench, the bench top itself weighs a half a ton. And each of the legs have airbags in them. So you got a half a ton floating on air. Does a pretty damn good job. Yeah. The one environmental interference that we get are cosmic rays. The photodetector is so sensitive that, I don't know what the frequency is, but it's not uncommon to find a spike in the spectrum that isn't where you predicted, and it's a single pixel wide. And that's a cosmic ray coming through the building and through our detector. And the software is a routine that scrubs out cosmic ray interference. That's how sensitive the photodetector is. And it has to be to make these kinds of measurements, such a weak phenomenon.
So I have a question, like there are many bacteria that cannot be grown in the lab. So like, can you use the result of like grading or any like other way to like predict or build model on those like bacteria that cannot be grown in the lab?
So you can do experiments in the field with stable isotopes, preserve them, and then do surveys of the community. See how active they were, and then you can look at their predators as well to see if they inherited the isotopic signature of their prey, but it's really challenging. Yeah. You know, I kind of sugar-coated it, 2 seconds per spectrum, et cetera. But there are things that interfere with the analysis. It's not always 2 seconds you've done. Sometimes it's a minute. Sometimes you have to pre-treat the samples because they fluoresce. Now, fluorescence is a useful tool in some respects, but fluorescence is our enemy when we're doing Raman spectroscopy, because the fluorescent peak is very broad and it hides all the information that's underneath from Raman scattering. But we've devised ways to counteract that. Hey.
Yeah, thanks for the talk, really cool stuff. For determining the single cell growth rates, how do you determine which Raman band you're gonna monitor? And like in the case of the carotenoid pigments, you said there's heterogeneity in single cell growth rates. How do you know it's that and not heterogeneity in carotenoid content of individual cells?
Well, the frequency shift is independent of the content of carotenoid content.
Right, but you're presuming that as there's more, as you've got more carotenoid than C13, that you're making the assumption that that is then directly proportional to the overall cell growth rate. Like, could you have a cell that just like grew more carotenoid in it?
That's possible, and we have a way that we can test that, but and we're not the only lab that's made these kinds of measurements, and there's some pretty good evidence that they're in lockstep. The carotenoid production rate and the division rate are in lockstep. What was the other part of the question?
Oh, kind of the first one, why are you monitoring carotenoid?
Yeah.
One for another one, you're monitoring phenolalanin.
Right. Okay. So with photosynthetic organisms, although we're finding that from some of our field samples, the carotenoids aren't working out so well, right, Natasha? Yeah. In the lab, they work great. So carotenoids, the reason to choose them is because if you excite them within their resonance band, so within the electronic transition band, you get much higher scattering efficiency. It's called resonance Raman, and you can get signal amplifications of about 1,000-fold compared to spontaneous Raman. So we use a green laser, which excites carotenoids within their optical absorption band. And that's why we get information in 2 seconds. With non-photosynthetic organisms, we pick phenylalanine because, if you recall from those spectra, it's a really, really sharp peak with very few interfering peaks near it. Whereas if we picked peak that's indicative of lipids or something else. There's a lot of other peaks in close proximity. So it's pretty unambiguous when you look at phenylalanine. So we can actually, and Natasha's looking at this, photo bleach the photosynthetic organisms and look at their phenylalanine peak as well. So that your first question, we could actually evaluate, if we had the time and money, we could evaluate that by looking at carotenoids and phenylalanine on the same cells.
Right. So you suggest that through the single cell growth rates that each cell has a different growth rate slightly. I assume this is destructive, so the cell dies after it dies. But
Well, we preserve them before we even look at them.
can you,
If they sat still, we could look at live cells.
could you then select the ones that are growing faster according to your measurements and then cultivate them and have two different cultures, basically one growing very fast and one growing slower?
Yes.
And if so, it seems like that might be something really exciting and could apply it to many different systems.
Yeah, my colleagues slash competitors in Zurich and Vienna have developed an instrument that is akin to a flow cytometer where they do Raman cell sorting. And so you have live cells in a microfluidic stream, and they've labeled them, I think they use deuterium, but to give you C13. As they pass through the beam, they recognize isotopically heavy versus isotopically light. If they're isotopically heavy, they go in this direction to the cell sorter. If they're isotopically light, they go into the discard bin. They got it, they built it, it's the only one in existence. There's a Nature paper that I reviewed 3 years ago.
You should have given the talk earlier, you could have.
Yeah, I mean, another $500,000 and I think we can build it. Any other questions?
Alright, there is
Any online?
none. Yeah, no, I opened it. I think there are
Oh, you
people
did?
online,
Okay.
but there's no questions. But there is lunch, so come join us for lunch. Take the opportunity to talk to Gordon. Maybe you have some specific research questions that could be addressed with this technique. Take the opportunity.
Techniques, yeah.
And thank
And-
you. for
Techniques. And yes, thank you.
We can arrange a tour. It's very beautiful.
Come take a tour.
Thank you, Gordon.